Hello, everybody. My name is Justin Dr. Chun, and I'll be moderating today's session. And I'm an assistant professor and nephrologist at the University of Calgary. Just to begin this session, we'll do the land acknowledgements for the University of Alberta. And this is in the Treaty 6 territory. And we'd just like to respect the history, languages, and cultures of First Nations, Métis, Inuit, and all the First Peoples of Canada, whose presence continues to enrich our vibrant community. And thanks to our sponsors, Paladin, Astellas, AstraZeneca, for their support of the ATI seminar series. And just a little bit of the bio about Dr. Lakshman Gunaratnam. He's a nephrologist and associate professor of medicine at Western University. He currently holds the Robert Zhang Chair in Translational Transplant Research at Western and is the medical director of multi-organ transplant program at the London Health Sciences Centre. He completed his undergraduate and graduate studies at the University of Toronto in immunology, and then went on to attend medical school and completed residency training in internal medicine at the University of Ottawa. He completed his research fellowship at the University of Ottawa and Harvard Medical School in cancer biology and acute kidney injury, respectively. And after completing his clinical fellowship in nephrology at the Mass General Hospital and the Brigham and Women's Hospital in Boston in 2009, he did additional training in renal transplantation at Brigham and Women's Hospital. He was promoted to instructor in medicine at Harvard Medical School and later served as an associate physician at Brigham and Women's Hospital prior to moving to London, Ontario. He leads a basic science and translational research program focused on the biological mechanisms, understanding acute kidney injury, prolonging the lifespan of kidney transplants, and novel treatments for kidney cancer. His research program is supported by peer-reviewed funding from several agencies, including CIHR, the Kidney Foundation of Canada, CIRC, the Government of Ontario, and the Canadian Foundation for Innovation. He also holds several advisory and leadership positions in Canadian medical organizations, including Canadian Blood Services, the Kidney Foundation of Canada, Trillium Gift of Life Network, Canadian Society of Transplantation, CIRH, and the Ontario Medical Association. And he's an amazing colleague, and I've seen him present many times too. And I'm looking forward to this talk, which is entitled The Multifaceted Role of KIM-1, TIM-1 in Renal Transplantation. Thank you, Lakshman.
Justin, thank you very much for the kind words. And I would really honor to be presenting here. I also want to acknowledge the lands that I'm standing on, the First Nations land, and I will start. So I'm going to talk to you about my passion, which is KIM-1 or TIM-1, and I'll explain this confusion in a moment. So here are my conflicts and the objective which were handed out. We'll be following them in no specific order, but we will cover them all. So just to start off, this is a basic science talk, but ultimately we're trying to help patients. And really in my business, I want to help patients with end-stage kidney disease. And we do believe that transplantation, kidney transplantation, is the treatment of choice because it improves survival and quality of life for our patients with end-stage kidney disease. However, I think you guys are no stranger to understand the survival benefit of transplant. And here's a picture I'm not sure if people recognize who this is. Selena Gomez receiving a transplant. It's wonderful. However, to this day, we face many barriers to successful transplantation. And obviously, the first one is the availability of organs. And then the alloimmune response continues to be a major impediment to successful transplant. And really, when I mean by successful transplant, is really one transplant for life, really, for a patient. So they don't need retransplantation. Non-alloimmune injury, such as ischemia-reperfusion injury, is also a major clinical problem in transplant. And then, of course, the side effects of immunosuppressive medications. I would love for my patients not to be on immunosuppression. However, for today, we're going to focus on two areas, which is the alloimmune response and the ischemia-reperfusion injury as it pertains to KIM-1. So to explain this molecule, KIM-1 and TIM-1, I'm going to give you a bit of a history. KIM-1 was originally cloned as the hepatitis A virus receptor. This is the virus, one of the most important viruses in the developing world. and this is thought to be the receptor. And so therefore it was named as HAVCR1, and it was originally identified in monkey kidney cells. Then the laboratory of Bonaventure et al. Identified a molecule known as KIM-1, which they found to be upregulated on proximal tubule cells during kidney injury and repair, and they began studying that. Shortly thereafter, the KIM-1 gene was cloned and was found to be identical to HAVCR1, and therefore it was actually the same gene, only they had given a different name. At the same time, the group of McIntyre, Umetsu, and so forth were hunting for a new gene that determines susceptibility to asthma. And they stumbled on a family of proteins or genes called as TIM proteins, the T cell immunoglobulin and mucin domain containing proteins. And when they sequenced this, they identified that the gene sequence was identical to hepatitis A virus cellular receptor. And basically, these were the same gene. And later on, it was shown that genetic variants in TIM-1 also predicted susceptibility to allergy in patients who had been exposed to hepatitis A virus. So the gene transcript for hepatitis A virus is present in lymph node, kidney, and then also in spleen and lung and thymus. And the highlighted blue topics will be discussed in a minute. Now, at this time, I was just arrived in Harvard Medical School to do my postdoc on KIM-1. And then this is how I felt right after what happened. The immunologist decided to call this molecule TIM-1, so everybody started calling it TIM-1. So it felt like they changed the name Kim's convenience to Tim's convenience. So what is this TIM-1 molecule? Let's call it TIM-1 for the moment to make the immunologist happy. It is a receptor, a cell surface receptor for phosphatidylserine. Phosphatidylserine is a marker that's found on, it's a phospholipid found on the outer surface of apoptotic cells. So here's a picture of an apoptotic cells, and they're found on the outer surface, and it's thought to mark them for removal by the body. And the KIM-1 or TIM-1 molecule is a type 1 transmembrane protein, which is made up of the IgV domain, mucin domain, the sugar domain, and then the cytoplasmic tail in a transmembrane domain. What is unique about KIM-1 and TIM-1 is that they have a unique structure with highly conserved cysteine residues that forms a pocket that allows TIM-1 to recognize phosphatidylserine, which is its major natural ligand. And this is highly conserved among other family members of the TIM family members I mentioned, and among different species. So let's talk about the alloimmune response, which I told you is one of the major barriers, and the role of TIM-1 in this process. In order to talk about this, I need to introduce you to regulatory B cells. So B cells are B lineage cells produce antibody, and they partake in humoral immunity, and we're well aware when this goes wrong in transplant rejection, so antibody-immune rejection. But importantly, B cells are also very powerful antigen-presenting cells, and they can both co-stimulate or co-inhibit T cell responses and regulate immune responses. Now, regulatory B cells are a very small subset of B cells that actually regulate immune responses, and they've been implicated in regulating autoimmune disease and preventing rejection. The B-reg activity, its propensity to modify the immune system is attributed to IL-10 production, but also other mechanisms that we may discuss briefly. At this point, there was no specific markers. We know they existed, but there was no specific marker to identify these B regulatory cells, and if they even were important in the first place. And this takes us to work done by Dr. David Rothstein at the University of Pittsburgh Medical Center. And he was the first to identify that TIM-1 was expressed on B cells. Until this time, there were many, many papers from a number of labs showing that TIM-1 may be on T cells and may be regulating T cell response in asthma. But many people have a hard time finding TIM-1 expression on T cells, let alone human T cells. He identified that up to 5 to 8% of B cells constitutively express TIM-1. And this you can see here on flow cytometry as well as Western blot. And this had never been shown before. And you can see clearly it was only detectable in the B cells by Western blot. Now, what was dramatic here is that Dr. Rothstein's group showed that when you stimulate B cells with the anti-TIM-1 antibody, you can actually prolong islet allograft survival. So this is a islet allograft model, and here is the survival for the controls. However, in the animals that were stimulated with the anti-TIM-1 antibody, which is known as RMT10, basically it's an anti-TIM-1 antibody, you can see that you can dramatically prolong the survival of these allografts. Now, this was lost if you depleted B cells with the anti-CD20, suggesting that it was B cells that were responsible for this prolongation. And the reverse, he showed that if you take a B cell deficient mouse and transfer them with B cells and then stimulate them with the anti-TIM-1, you could get the same result, suggesting that it was B cells that were doing this. Now, in addition to that, you can see that the B cells, when stimulated with TIM-1, or when they undergo transplantation with the islet, you can see that the B cells start upregulating TIM-1 levels just with transplantation of the islets. But then if you stimulate in addition with anti-TIM-1 antibody, you can see this dramatic upregulation of TIM-1 on these B cells, CD19 positive B cells. And then when you stimulate them with the anti-TIM-1 antibodies, you make these B cells produce interleukin-10 and some IL-4. But also, if you purify just the TIM-1 positive B cells, you can see that majority of this IL-10 and IL-4, which are Th2 and immunosuppressive cytokines, are coming from these B cells. And of course, if you do the same islet transplant model and stimulate with anti-TIM-1, but in an IL-10 knockout or IL-4 knockout background, you can see that you lose the ability to prolong transplant, suggesting that anti-TIM-1 works through the mechanism requiring IL-4 and IL-10. And so therefore, the B cells were producing these cytokines, which promoted tolerance here in this model. So basically what Dr. Rothstein is proposing, that anti-TIM-1 antibody would stimulate B regulatory cells and we're calling them B regulatory cells because they're modifying the immune response by definition. You can see that they upregulate TIM-1 expression, and then they also modify by cytokine environment producing IL-10, and also skewing the T cell response towards a Th2 response. This brings me to Dr. Vijay Kutra, who's a preeminent immunologist at Harvard Medical School, who is working upstairs to where I was doing my postdoc, who at the same time, and often in collaboration with Dr. Rothstein, generated what is known as the TIM-1 mutant mouse. And what his group did is they deleted the mucin domain. If you remember that sugar domain that I mentioned, they deleted this domain in mice. To be honest, this was actually accidental, but it was important because this area was variations in this area were responsible for the association with allergy and hepatitis A. So they wanted to know, does this actually affect the protein function? So they made a KIM-1 delta mucin domain mice, which lacked the mucin domain, which is a shorter KIM-1 protein. And then they observed that these mice naturally, as they got older, started losing IL-10 producing B cells. And we suspected these were the B regs because they were already identified by Dr. Rothstein. But the mucin domain deletion mice, of course, have very few B reg cells. And this became worse as the mice got older. And in association with this, these mice that had the defect in B regs here also had enhanced activated T cells, especially secreting interferon gamma and IL-17 in correlation to this phenotype. Now, what's interesting is if you follow these mouse out to older age, up to 18 months, you can see that they naturally develop splenomegaly or enlarged spleen, and also they get autoimmune phenotype, which is inflammation in multiple organs, including the liver and the lung. And again, you can see that the cytokine profile of the TIM-1 mutant mice, here it's a TIM-1 knockout mice that they had also acquired in the study. And you can see that the cytokine profile is dramatically skewed in the TIM-1 deficient mice. Now, this is a multiple sclerosis animal model known as EAE. And in this model, the mice develop what appears to be like a multiple sclerosis-like phenotype. And what they did here is they transferred wild-type B cells or B cells from a TIM-1 knockout mice. And you can see that when you transfer these B cells, you can dramatically ameliorate autoimmune disease, very much like the B regs that normally do this. But here, these are TIM-1 positive B regs. And again, the alteration of phenotype was associated with reduction in IL-17 and enhancement of FOXP3 or T regulatory cells. So these B regulatory cells are very profoundly immunosuppressing this animal and reducing the severity of autoimmune disease by skewing the T cells that are causing the multiple sclerosis-like phenotype. Now, then this brings me to work under the same model here. Again, these are the TIM-1 knockout mice and the TIM-1 wild-type mice. And so I told you earlier that the mature ligand for TIM-1 is apoptotic cells, specifically phosphatidylserine on apoptotic cells. So here, Dr. Kutra's lab asked the question, do these, the TIM-1 knockout mice, do they bind to apoptotic cells? And obviously you can see on the right-hand side, this is a flow cytometry, and you can show on the right-hand side, in the red is the binding of apoptotic cells, which are fluorescent, to the wild type TIM-1 positive B cells. However, if you do not have TIM-1, you can see that they lose binding to apoptotic cells. So obviously, if you don't have TIM-1, you cannot bind to apoptotic cells. Again, this suggests that this is the major receptor for apoptotic cell recognition by these cells. And then these are B cells. And then you can see that when you stimulate the cells with apoptotic cells, which has really never been shown here, it leads to interleukin-10 production, and like we saw earlier in the B regs. And this is lost upon TIM-1 deletion. So if you get rid of TIM-1, the B cells are no longer able to respond to apoptotic cells, and most importantly, produce IL-10. And again, here you can see if you transfer this mice with the TIM-1, so if you take the TIM-1 knockout mice and the wild type mice, and then you use the EAE model to induce multiple sclerosis, you can see that if you stimulate with apoptotic cells in the wild type cell, just stimulating with wild type apoptotic cells, you can see that you can reduce the severity of EAE, which is experimental allergic encephalomyelitis or multiple sclerosis. But if you take the TIM-1 knockout mice and stimulate them with apoptotic cells, it doesn't happen. This was pretty dramatic, suggesting that TIM-1 can cause immunomodulation by apoptotic cells. Now, this brings me back to Dr. Rotstein's work. Now, he, at the same time, around exactly the same time, Dr. Rothstein showed that again, the mucin domain mutant mice and the wild type mice, he shows here that if you let them bind to phosphatidylserine, bind to liposomes, you can see that the wild type B cells can bind to the liposomes coated with phosphatidylserine. However, the mutant mice cannot recognize it. Here's a picture showing how a B cell that expresses TIM-1 in red here is binding to apoptotic cells. Now, when you stimulate the B cells with apoptotic cells, you can see that they start expanding, just like the first figure I showed you, is that when you stimulate them with apoptotic cells, you turn on these IL-10 producing B regulatory cells, and they dramatically expand, and with the more apoptotic cells you stimulate them with. So basically, instead of using anti-TIM-1, now they can use apoptotic cells to stimulate the production of IL-10 by these B regulatory cells. And of course, how is this relevant to transplant? So here he used a cardiac transplant model, and he shows that if you do a heart transplant, which is a B6 into BALB/C heart transplant into B cell division mice, and then you transfer either wild type B cells or the mucin domain deletion B cells, and then stimulate them with anti-TIM-1 antibody, you can see that the wild type animals, you can prolong the survival of heart transplants. However, in the mucin domain deletion, even if you stimulate with anti-TIM-1, it has no effect. And again, this is associated with IL-10 production, which is dependent on anti-TIM-1 antibody. So either apoptotic cells or anti-TIM-1 can turn on B regs, and then they can protect from heart transplant rejection. Now, then we go to the TIM-1 mucin domain mice, and then if you do a heart transplant directly into them without any anti-TIM-1 stimulation, you can see that the wild type mice which are able to prolong heart transplant. However, the mice that have mucin domain deletion of TIM-1, you can see that they lose their heart transplants very quickly, suggesting they've lost some kind of tolerance ability that was naturally there. And then this he reproduced by using adoptive transfer. So basically, you can transfer the B cells back from the wild type or the mucin domain, and you can see that only the wild type B cells are able to rescue heart transplant prolongation, whereas the TIM-1 negative from the mucin domain mice cannot do that. Okay, and if you take a TIM-1 mucin domain deletion mouse and then give it back normal B cells, you can see that you can rescue that, suggesting that it's the TIM-1 positive B regulatory cells that were responsible for prolonging the transplant lifespan. Now, you might ask, okay, this is all wonderful. We're just talking about mice. What about humans? Does this even matter in humans? Obviously, we're very limited in the type of transplants we can do, but in this study by Catherine Wood, I think it was a major study, because until this time, nobody had ever shown that these B regs, especially TIM-1 positive B regs, existed in humans. And so here she stimulated B cells, CD19 positive B cells, with CD154, which is also the CD40 ligand, which is an important ligand for CD40 on T cells. But here, the CD40 ligand is used to stimulate B cells. And as the B cells expand, they start producing IL-10, very akin to the B regulatory cells that produce IL-10. But in addition to producing IL-10, you can see that stimulation with the CD154 leads to not just IL-10 production, but also TIM-1 production, which means these B-regs are producing TIM-1 and producing IL-10, and therefore this is thought to be a marker for B-regs now. And then here in a skin transplant is a humanized mice where they did skin transplant. So this is really very cool. And they showed that if you do a skin transplant and you transfer in the B-regs that were expanded with anti-CD154, which produce TIM-1 positive B-regs, you're able to prolong skin transplant. However, in the control situation, you do not have that. Okay. And then and this is also correlated with the in the animals that you have expansion in the skin of these immunomodulatory or B regulatory cells in the actual graft. So just that the B regulatory cells are infiltrating the graft and prolonging the lifespan of the graft by reducing the rejection of these skin transplants. So let's put this all together. Okay, so here you have B cells that have small amounts of TIM-1 on their surface, but upon stimulation with transplant or some encounter with the B cell antigen, you get upregulation of TIM-1. And this one can be further enhanced by either anti-TIM-1 antibody or apoptotic cells, which leads to not just upregulation of TIM-1, but also expansion of the B-reg population. You saw that there was a massive increase in the B regulatory population when you do either of these things. And when this happens, through, and I didn't talk about this, but through mechanism involving TIGIT and other proteins, these can directly regulate the T cell responses, skewing it towards the Th2 and Tregs, and also down-modulating Th1 and Th17, which are important in rejection and other autoimmune diseases such as multiple sclerosis. So these are very much like T regulatory cells, and these are B regulatory cells, and they sense the environment for dead cells using TIM-1. Now, just in a complete aside here, I just want to show you that it's not just B cells. It is also now known, this is work from Dale Emutsu, who originally was one of the groups who identified TIM-1. They showed that NKT cells, which are normal innate like T cells that have important also in transplant and other diseases, naturally express TIM-1 on their surface. And when you stimulate these NKT cells with alpha-gal-Sur, which is an antigen that causes stimulation of NKT cells, in addition to anti-TIM-1 that we talked about earlier, you can see that you can activate these NKT cells to produce cytokines such as interleukin-4. And if you took NKT cell deficient, you don't have that response, suggesting these were actually NKT cells producing it. What is more important is that if you stimulate these NKT cells with apoptotic cells, these are called eruptotic red blood cells, which basically mimic apoptotic cells. They have phosphatidylserine on their surface. You can actually mimic the anti-TIM-1 stimulation just like in B-regs. The only difference here is it's causing it to produce something more dramatic, which is airway hypersensitivity. If you remember, I did tell you that originally that TIM-1 was linked to allergic responses. So what they did here is they created an allergy model. So it's basically they give intranasal ovalbumin to the mice to create some kind of like an asthma model. And then they measure the resistance in the airways, and you can see that when they stimulated, here's the control here. If they the controls are right here. If you stimulated the NKT cells with alpha-gal-Sur, and you can see that you can enhance the response. But however, if you stimulate it further with eruptotic red blood cells or apoptotic cells, you can see that you can enhance the airway hyperactivity. In fact, it makes asthma worse. So unlike B-regs, which suppress immune responses like Th1 and Th17, these NKT cells, when stimulated with apoptotic cells, they actually make airway inflammation worse and airway hyperactivity worse. And that is because the cytokines that are involved here, like Th2 cytokines, are making allergy worse here. So to summarize this part, TIM-1 and KIM-1 are the same molecule, only they have different names, but they both bind to phosphatidylserine, and because they're phosphatidylserine receptors, they're encoded by the exact same gene, which is due to history known as HAVCR1. TIM-1 now is recognized as a broad and important marker of B regulatory cells that produce IL-10 in both mice and humans, as I showed you. TIM-1 allows cells that express them, including NKT cells and B cells, to recognize apoptotic cells in the environment, which could be from injury, inflammation, and so forth. and then regulate alloimmune responses in the case of Bregs, but in the case of NKT cells, they make asthma worse. So it's not always a good thing as far as patients are concerned. Now, bringing it back to KIM-1, which we're going to talk about next, TIM-1, when it's expressed in injured proximal tubule cells, we call it KIM-1. But please remember, they're exactly the same thing. The only difference is when we talk about the kidney, we like to call it KIM-1. And back to my favorite show, which is Kim's Convenience, because now we're back to calling it KIM-1. So we're going to switch gears now to talk about KIM-1 in renal transplantation. Now, in order to understand KIM-1 in renal transplantation, we need to understand ischemia-reperfusion injury. Now, delayed graft function is when, after transplant in a human, the kidney transplant doesn't work very well, and often you need dialysis in the first week after transplant. So this is kind of functional readout. But really, what causes this to happen when we do transplant is ischemia-reperfusion injury. And that is when you have reduced blood flow to the organ during the procurement and transplant procedure. And when it's severe, it can cause organ dysfunction, otherwise known as delayed graft function. This delayed graft function is an expensive problem, and it also leads to poor long and short-term outcome for the patient. So it's an absolutely important problem, as I highlighted to you in the first slide when we talked about the major barriers. Now, delayed graft function is differently associated with different types of kidneys. Like if you have a living donor, we have very little of it. If you have a deceased donor with a donation after cardiac death, this is the old cardiac death donors, or donation after cardiac death, you have much higher levels of DGF. And in London, we do lots of these, so it's a big problem for our patients. Now, a little bit about pathophysiology of ischemia-reperfusion injury. Again, ischemia is a reduced blood flow or lack of blood flow, and then reoxygenation or reperfusion is when the blood flow is reestablished. The problem when you have reduced blood flow is you have decreased oxygen supply to the kidney, and the kidney, unfortunately, is not very good at glycolysis, and therefore it relies on oxygen and also beta oxidation, which is using fatty acids for its metabolism. So when there's decreased oxygen supply, these tubular cells are not very happy, and they start dying and they're causing inflammation. And the cell death is due to the lack of ATP, but then also you get the wave of reactive oxygen species that happens when, of course, you reestablish blood flow. This could be a situation where the kidney is on ice, and you put it back into the recipient and sew everything together. Whoosh, you get a warm rush of warm blood. We call it warm ischemia. And this whole process of ischemia and extra oxygen causes DNA damage and can lead to apoptosis or regulated necrosis. And this leads to inflammation. Now, why is necrosis so bad? We always talk about necrosis, and the difference between apoptosis and necrosis, one of the main important things is under necrosis, the membrane integrity of the cells that are damaged is broken. And when that happens, the cell becomes leaky, and it starts leaking out intracellular contents, which is supposed to be hidden, into the extracellular environment. And this triggers inflammation, not just in the kidney, but even beyond the kidney and the immune cells, through receptors known as danger receptors. The most famous of them all is toll-like receptor. And so cells that are in the surrounding area around the dead cell, they get inflamed, and we call this necroinflammation. Now, why was this all important in transplantation? I apologize, I had a liver here, but I love liver transplants as well. And here is a liver that sustained ischemia-reperfusion injury. And these star-like things is all these danger molecules leaking out of the dead cells. And when they leak out, they can activate, for example, an antigen-presenting cell, which is sitting there, to become more inflamed and become more adept at triggering rejection or allo stimulation, and then therefore activating T cells more. And then when T cells get activated, the alloreactive T cells, they come back and destroy the graft. So ischemic injury, we believe, can potentiate alloimmune responses and rejection responses, and it makes the whole thing a big mess, really. Now, let's come back to remind you about KIM-1 now. They talked about ischemia. Again, KIM-1 is encoded by the HAVCR gene. It's a type 1 transmembrane protein. It binds to phosphatidylserine. And here it is, a picture of it on the surface of a membrane here. Now, what's important about KIM-1, it is specifically expressed in the injured kidney, but not the healthy kidney, in the proximal tubules, but on the apical membrane. So this is the single tubule, and this is the lumen, where the ultrafiltrate runs. And so KIM-1 is produced at very high levels after injury, but not in the healthy condition. And some of it is in fact shed, and we can detect this, and it's a marker for acute kidney injury for clinical purposes. Now, at the molecular level, many years of work, both when I was at Harvard, but also here, suggesting what does this actually do? So in T cells, it seems to be a sensor for apoptotic cells. What does it do in tubular cells? So when tubular cells express KIM-1, it transforms these proximal tubule epithelial cells into phagocytes for removal of these dead cells. So apoptotic cells that bind, that are dying left, right, and center from the ischemia, they bind to the apoptotic cells here. The KIM-1 binds to apoptotic cells through the phosphatidylserine with the little triangles. But here, instead of just reacting to it, it actually eats them. And you can see pictures of this from Dr. Bonaventuri's group showing that here's red KIM-1 in a rat kidney proximal tubule, and these dead cells being eaten while the kidney is injured. And so basically, you can see here, here's a single cell we took a picture of in the test tube, which expresses green KIM-1. And as soon as you feed it an apoptotic cell, which is in red here, you can see that the KIM-1 grabs onto it, just like this cartoon. So it's not a lie, it's really happening. It grabs onto it and swallows it up and eats it and removes it from the extracellular milieu. Now, we have shown that KIM-1 is absolutely crucial for removing dead apoptotic cells from the kidney, because if you isolate proximal tubule cells from KIM-1 knockout mice and wild-type mice, and you feed them fluorescent apoptotic cells and ask them to see how well they eat, you can see that KIM-1 wild-type can eat the dead cells very nicely. But if you don't have KIM-1, you can't do it very much at all. And which means that KIM-1 is a major phagocytic receptor in the kidney, especially after injury. So what we hypothesize is that here's your normal healthy KIM-1 wild-type mice or humans, and when they have ischemic injury, they have a bunch of dead cells, including apoptotic and necrotic cells. And apoptotic cells, they can also become secondary necrotic because they can slowly dissipate and start falling apart and become necrotic. And obviously, I told you earlier that necrotic cells are bad because they start leaking stuff like HMGB1, which is a danger molecule, out of theirs, and they will stimulate inflammation in the kidney. But guess what? If you have KIM-1 around, it can bind and delete and start engulfing these dead cells, removing them from the kidney and reducing inflammation. But if you don't have KIM-1, all this gunk accumulates and causes inflammation. So the question is, what happens in a kidney transplant? Is KIM-1 even important in kidney transplant? So we turn to a Syngenic murine kidney transplant model. And what this is basically they're genetically identical recipient and donors, B6 to B6. The only difference is we took kidneys from KIM-1 wild-type mice and KIM-1 knockout mice, and then we transplanted them into wild-type B6. So these guys are, these mice have normal KIM-1, but the donor kidney either has no KIM-1 or KIM-1. And then we put them into these mice, and it's a life-saving kidney transplant because these mice's normal kidney is removed and they get a kidney that they inherited from one of the donors. So either they rely on KIM-1 knockout or wild-type kidney for survival. And this is work we published some years ago, but this is work by Ji-Yoon Lee, my PhD student. She's at Merck now. And so basically she showed that as soon as you transplant, you can see KIM-1 in the kidney. It's expressed in the kidney very soon after transplant and persists over time. And so KIM-1 is induced, just like we said, in the ischemic injury, but only here in a transplant model. Now, what's important is, does it really matter? So what about graft function? So you can see that after transplantation, the kidney function of the mice that have a normal KIM-1 is wonderful after day 3, but the KIM-1 knockout mice have very bad kidney function. In the humans, this would be what we potentially call delayed graft function or kidney dysfunction due to ischemic injury. And you can see that when the injury is bad, obviously some mice died. And there's a survival advantage to having KIM-1 after transplant, because the mice that had no KIM-1 in their kidneys died because they needed that kidney for survival. So these mice unfortunately passed away. Some of these mice died. And so KIM-1 is not only important for good graft function, but survival after a kidney transplant in this model. Now, in association with the improved graft function and so forth, you can see that there's more inflammation in the KIM-1 knockout kidneys compared to the wild-type kidneys, and also more inflammatory cytokines. So the kidneys are inflamed. And more importantly, when we look at the dead cells, so I told you, if you're clearing dead cells and you don't have KIM-1, there should be a ton of dead cells around. And not surprisingly, we saw tons of apoptotic cells in the knockout compared to the wild-type because they are not being cleared in their knockout. And what was most surprising, there was so much necrotic cell death going on in the KIM-1 knockout mice compared to wild type. And this is not due to the fact that they're dying more. We've shown that in the paper, but it's really because you're not clearing them. It starts accumulating in these. Now, what was a bit surprising is the amount of necrotic cells in this situation. And so far, I told you that KIM-1 and TIM-1 only recognize apoptotic cells. And here's a nice old data from our lab showing that when you feed proximal tubule cells apoptotic cells, you can see they're able to phagocytose very well. So this is how much they're eating the dead cells. They eat the dead cells very well. And if you don't have KIM-1, of course, there's no eating. But when we fed them necrotic cells, whether you had KIM-1 or not, they were not eating, they ate a little bit, but not much. So the problem here is, okay, they can eat apoptotic cells, but who's clearing all the necrotic cells? We know for sure that KIM-1 is important because when we knocked out KIM-1, the transplants did poorly. So how do we explain this? This led to a collaboration with Dr. Miyazaki at the University of Tokyo, who was working on this molecule known as AIM, or CD5 ligand. And this is a protein that has been previously shown to be important for macrophage survival, but it's found in blood. This protein is a small protein, 38 kilodaltons. It's bound to IgM, which is an immunoglobulin in your body, and because it's bound, it cannot leave the circulation. Now, when kidney injury happens using ischemia, like AKI or acute kidney injury, these AIM molecules are able to leave the circulation because they're filtered in the kidney, and then they go into the kidney and start coating all the necrotic cells. And when they coat the necrotic cells, then now KIM-1, which was never able to bind necrotic cells, is now able to eat necrotic cells. This is just a cartoon. I'll show you the proof here. So here's proximal tubule cells that either express KIM-1 or don't express KIM-1, and we fed them necrotic cells. And when we fed them necrotic cells, in addition to AIM or recombinant AIM, they were able to eat necrotic cells very well, but in the absence of AIM, they cannot eat it very well. So what I'm trying to tell you is that KIM-1 can eat apoptotic cells just fine, but in order to eat necrotic cells, they need the help of this opsinin, and opsinin is a molecule that allows something else to eat it better. It's like putting salt and pepper on your food. It tastes a lot better. You put salt and pepper or AIM, and suddenly KIM-1 can eat the necrotic cells. Now, this is happening in all the wild-type mice. We just didn't know that this was the mechanism. So KIM-1 is important for both eating apoptotic cells and necrotic cells with the help of AIM. Now, AIM is wonderful because one of the biggest problems in transplant right now is delayed graft function. I told you that, and it's an important clinical and a healthcare problem. So what we want to know is, can we give extra AIM and improve delayed graft function, at least in mice? And so what we did here, we did the same Syngenic kidney transplant model, and then we gave the mice either PBS or recombinant AIM intravenously. We know it would be filtered because it's injured, the animal. And you can see it's quite dramatic. One single dose of recombinant AIM just at the time of transplant was able to improve kidney function right after transplant here. And in association with this, also decrease the tissue damage from ischemia. So recombinant AIM seems to be like a therapeutic for DGF, and it works through KIM-1, and that is already published, but I didn't show you that here, but it requires KIM-1, but it's a partner to KIM-1. So basically, what is KIM-1 doing is, we think that, obviously, I told you earlier that when you have injury, ischemic injury after transplant, you get all the cell death and all this necrotic cell death and all the stuff leaking out of there, which is going to stimulate the alloimmune response by stimulating antigen-presenting cells that are coming from the transplant and then turning on T cells. However, the natural defense mechanism of the body and the kidney is that it expresses KIM-1, which is rapidly removing all these dead cells, necrotic cells and apoptotic cells, and thereby reducing the inflammation in the kidney. And we presume that maybe it reduces the alloimmune stimulation. TIM-1 and KIM-1 are the same protein and encoded by the same gene, and they both bind to phosphatidylserine on apoptotic cells. TIM-1 is a marker of IL-10 positive B-regs, and they're very powerful and important in promoting allograft tolerance. So we all have it. When we have a transplant, these B-regs are there to help us. And if you deplete them for whatever reason, you can lose the protective effect of B-regs that is naturally present. And I showed that in the animal models. KIM-1 or TIM-1 is upregulated in kidney syngeneic grafts due to IRI and mitigates inflammation and secondary tissue damage. So if you had a syngeneic, just ischemia, if you just looked at the ischemia, there's no question that KIM-1 is protective, and it does so by clearing all these dead cells. However, based on data I presented today, which is unpublished, KIM-1 is persistently upregulated after allotransplantation. We're not sure why, but we do believe it's due to ongoing alloimmune injury. So in addition to ischemic injury, you have alloimmune injury attacking the tubular cells, which are leading to persistent upregulation of KIM-1 that normally doesn't happen. And this paradoxically seems to promote graft loss instead of survival. And so this is going to take some years to figure out, but we're super excited about that. I want to end by thanking ATI for inviting me to speak today, but also important people in my lab, which includes Elena Tutina-Fatan, who's working on the fibrosis story, Dr. Ole Ismail and Ji-Yun Lee, who did all the transplant work and the phagocytosis work, and Shobita, who's doing the she's actually working on the allotransplant model, and she's doing a PhD right now. My collaborators, especially Dr. Jenny Zhang, who helped us with the transplant model. My lab, and these are my two daughters, who, other than, they think KIM-1 is my life, but they are my life, to be honest. And thank you very much.
Dr. West, Laurie West.
That was wonderful, Lakshman. I'm sitting here in Union Station in Toronto. And it captured my entire attention. So I hope there are no announcements in the background. That's so much really interesting data. What does it say? Lower my hand. Anyway, so I don't even know where to start with questions. You'll see that Anne put in a question, which I'll just read for her because she had to leave quickly. She's wondering if you have data for this in an ABO incompatible transplant model. You knew I'd have an ABO question. Do you have such a thing?
No, we haven't. Clinically, obviously, you know, London, we do a lot of ABO incompatible transplants, but I haven't moved this question to the lab yet because we still haven't figured out what really KIM-1 is doing, obviously, in transplant model. But it would be interesting to know in situations where an additional immune injury, for example, from an ABO incompatible antibody, what it would do. Great work, but probably a few years down the line before we even establish a model for that. But obviously, a very good question.
I wonder if we could think about doing that in an transgenic mouse model that we have. That might be something to think about.
Yes, absolutely. And you know, we're going to rely on your expertise to help us with something like that because you have years of experience here. And I think this is an important clinical area to move forward in basic research because it's the ABO incompatible transplant is blowing up in Canada in terms of in a good way, not in a bad way. So yes, absolutely.
So another question I have is there was evidence some years ago with when David Rotstein was publishing and other people on BREGs that there was an age-related and sex-related pattern to BREGs. I wonder if you've looked at that. I mean, in that very young children, or I've forgotten now whether they were basic models or clinical observations, had higher levels of BREGs, possibly. And I think maybe it was Tom Tedder who had some work in mice, for example. I'm not really sure, but is there anything more about an age-related or sex-related?
No, I think, so again, the B-cell work is all from Dr. Kutra and Dr. Rothstein, and they work often together. So until recently, they didn't even know these existed in humans, the KIM-1 positive or TIM-1 positive BREGs. They do exist in humans now. But what happens with age is that our propensity for autoimmunity increases because those TIM-1 knockouts and mucin domain mice don't get autoimmunity till much later in age. And in fact, many people didn't even pay attention to it because only Dr. Kutra's lab followed them for so long, they started getting them. And at that point, not having TIM-1 means you have fewer B regulatory cells and you're more prone to autoimmune disease. And so it's not that and not that they are expanding, but more importantly, that you need them even more at that stage in your life because you've had so many injuries and potentially exposed to autoantigens and so forth. So it is more important later part of age to have these B regulatory cells present to help you with the modulating immune responses. And it may not just be autoimmunity, it could be many other immune responses. And obviously in a vaccine setting, it may be the opposite, right? Nobody's looked at that yet, but in autoimmunity, you want to suppress it. But in a vaccine situation or cancer immunotherapy, and Dr. Kutra just recently published in Nature about this, TIM-1 positive BREGs play a bad role for the patient because they actually they thwart anti-tumor responses. So for tumors, BREGs are bad. For transplant, BREGs are good. So it's not a simple treatment for everybody. It's going to depend on what you have, what situation you're in. Good question.
Well, like so many other things where you've got the two sides of the coin of tumor versus transplant.
Absolutely.
And since I've got my hand up more than anybody else, what was my other question? You know, this paradoxic data that you showed at the end is really interesting. And is there a role as well for other organs than kidney to look at this?
So KIM-1 or TIM-1 is not expressed in other organs except potentially maybe lung. But so in the parenchyma, kidney is probably the only place. There was thoughts about liver, but in the mice, it's not there. We've looked for it. So, but other molecules that are important, other PS or phosphatidylserine receptors that are expressed, for example, in the eye and in the lung, might have a similar role, paradoxical role. Nobody's looked. but in the kidney, it is really quite shocking, and both in the transplant and non-transplant model. So I think this is not unique. It's something to do with chronic and persistent injury. It's bad news, and we know this is bad news just by looking at pathology and specimen. But now we might have a potential molecule that might be responsible for it.
And just one more question. Sorry, I'm badgering you with questions, but what about extracellular vesicles? What about the role of those? I mean, you talked briefly about necrosis and so on, but
so it's been known for some time that TIM-1 and KIM-1 can bind to extracellular vesicles because many of them do have phosphatidylserine on them. So we have not looked at it directly, but a few other labs are looking at this right now. And in fact, there have been a few publications where they've targeted for acute kidney injury extracellular vesicles loaded with microRNA or whatever it be your favorite target. You can deliver it to the kidney using KIM-1 or TIM-1 to the kidney and rescue AKI, for example. So people have used it as a therapeutic target. And that application can go to cancer, where KIM-1 is widely expressed in cancer. And I didn't present any of my cancer work today. As you know, my lab works on renal cancer. And in there, KIM-1 can be targeted as well. So KIM-1 is not just the biological important molecule, which I'm interested in, but it may be also a targeting molecule. So you can deliver goods using liposomes, for example. Thank you. Great questions, Lori.
What a treat to hear all this. And I'm the only one with my hand up at the moment, but it's just really wonderful work and so elegant. So thank you very much for speaking to us about this.
My pleasure. My pleasure.
I don't see any other hands up, but I did have a question, Lakshman.
Yeah.
The latest data is super intriguing. You talk about these macromolecules, phosphatidylserine, as well as extracellular vesicles. Joe Bonventre, he has a cell metabolism paper from 2021 that showed palmitic acid, and that being blocked in a diabetic mouse model with a TW37. Is that something that you've tested or something?
Yeah, so basically what he shows in a diabetic kidney model, he shows that KIM-1 can potentiate diabetic kidney injury because it allows the kidney to absorb palmitic acid containing lipids, which are pro-inflammatory and then make kidney disease worse. And then he shows that you can rescue that using this inhibitor and it turns out that inhibitor is not very specific to KIM-1. It doesn't block signaling. It seems to somehow interfere with potentially palmitic acid maybe binding to it. So it is not highly specific, but it was very dramatic in that paper where it blocked the diabetic injury but I'm not sure if it's truly KIM-1 specific, but it was dramatic, the results. You're right.
Are there any other questions? If not, we're approaching the hour. So in closing, I'd like to thank Lakshman in this amazing talk.